Hello, everyone. Welcome to the RADx Data Hub. We're having a webinar today talking about making data sets findable and accessible using digital object identifiers and other tools. My name is Susan DeBleek, and I am a research project manager for RENCI, the Renaissance Computing Institute. RENCI is one of the RADx Data Hub partners presenting today about the RADx Data Hub, and I'm glad to moderate the webinar today. The National Institutes of Health launched RADx, and RADx stands for the Rapid Acceleration of Diagnostics Initiative. It came from the urgent need to respond to the COVID-19 pandemic focused on diagnostic technologies. The RADx Data Hub is the infrastructure and the platform to house a number of studies associated with RADx. It's an aggregation of study data, including diagnostic developments, diagnostic deployments, and COVID spread assessments. So today we're going to be focusing on making RADx Data Hub study datasets findable and accessible. And today we're recording this webinar. We'll share the recording with webinar registrants in a couple days, and you'll also get copies of the slides. Today's agenda includes Anya Dabich will present on persistent unique identifiers. John Graybeal will speak about semantic identifiers and other IRIs. Peter Rose will present on compact URIs. And we'll wrap it up with questions for our presenters. Well, you all are familiar with Zoom, and just a reminder that we'll be using the question and answer function. So you should see two little bubbles next to each other that say Q&A. So use that part of Zoom to submit questions for our presenters today at any time, and we'll try to do our best to keep those questions for our question and answer session at the end of this presentation. So with that, I'd like to pass the microphone to Anya Dabich, and she will kick us off.
Thank you, Susan. Hello, everyone. My name is Anya Dabich, and I'm a lead scientist at Booz Allen Hamilton, one of the RADx Data Hub partners presenting today. To kick off this webinar, we will provide an overview of persistent unique identifiers and how they are used to support the research process by facilitating search, discovery, citation, and collaboration. Next slide. So a persistent unique identifier, referred to as a PID, is a long-lasting digital reference to a person, organization, or thing, which are used to unambiguously identify and access published content, usually by resolving to a URL. PIDs are globally unique, persistent, machine resolvable, and have an associated metadata schema that is retrievable using standardized internet communication protocols such as APIs. There are many different types of PIDs, but the three common PIDs used in research are digital object identifiers, used to identify digital objects, open researcher and contributor IDs, used to identify individuals, and researcher organization registry IDs, used to identify organizations. In the next slides, we will explore these different types of PIDs, their benefits, and how you can use PIDs to make your research outputs more findable, accessible, interoperable, and reusable. So digital object identifiers, or DOIs, are a type of persistent identifier used to uniquely identify research outputs, such as datasets, journal articles, books, and other digital objects that have been published and made electronically available. The benefits of using DOIs in research are that they enable other researchers to properly cite data and other research outputs, increasing the visibility of the original research. They also facilitate linkages between data shared through digital repositories and other related research outputs, such as articles and documents and datasets that have been published elsewhere, increasing transparency in research. And finally, DOIs enable researchers to comply with the NIH data management and sharing policy, which asks researchers to specify how their data will be made findable and identifiable, encouraging the use of PIDs to achieve this. In the future, DOIs will be assigned to studies shared through RADx Hub to facilitate proper citation and reuse. Next slide. And so how are DOIs issued? The publisher, such as a digital repository or a journal, will assign a DOI to the research output when it is published and made available electronically. These DOIs are minted by DOI registration agencies, such as Datasets, so that they are globally unique across all systems. And so researchers should use DOIs to properly cite published content. Next slide. The next type of persistent identifier is the Open Researcher and Contributor ID, or ORCID ID. And this is a type of persistent identifier that can be assigned to anyone involved in research, scholarship, or innovation, and are used to directly link authors to their research outputs throughout the course of their research careers. The benefits of using ORCID IDs is that they ensure unambiguous identification of the researcher. And this is important as many researchers may share the same or similar name. They may work or publish under different versions of their name, or they may change their name. ORCID IDs ensure the unambiguous identification of the researcher by assigning the researcher a PID. ORCID IDs also make it easier for others, such as potential collaborators or funders, to identify a researcher's contribution and to track research impact. And finally, since a researcher's biographical information is associated with their ORCID ID, this simplifies user registration within systems that are capable to extract information from ORCID.org through an application programming interface, streamlining the user registration process. In the next release of RADx Hub, users will be able to provide their ORCID ID during user registration. Next slide. So how to get an ORCID ID. Individuals interested in obtaining their own ORCID ID to uniquely identify themselves can register at orcid.org using the link provided here. Next slide. The last type of persistent identifier is the Research Organization Registry ID, or ROR ID. And this is a type of persistent identifier assigned to research organizations, such as universities, research institutes, labs, or any organization that's involved in scholarly research. The benefits of using ROR IDs is that they ensure unambiguous identification of organizations. And so like individuals, organizations may change names over time. And so PIDs ensure that organizations are identified consistently across systems. ROR IDs also enable researchers to link their research outputs to their affiliated organization, which streamlines the process of tracking research impact and identifying collaborations. And finally, ROR IDs also facilitate reporting on the outputs of institutional research, which is central to many research stakeholders. In the next release of RADx Hub, users will also be able to provide their ROR ID during user registration. Next slide. Individuals interested in creating a ROR ID to uniquely identify their organization can register at roar.org. If you are unsure whether your organization already has a ROR ID associated with it, you can explore registered organizations on the roar.org site. And both the ROR ID and ORCID ID are free for individuals to register. So in summary, PIDs establish connections between different entities within the research ecosystem via their underlying metadata schema, and they provide a unique, persistent, and long-lasting reference to these entities. By incorporating PIDs such as DOIs, ORCID IDs, and ROR IDs in their research, researchers can make their data more findable, accessible, interoperable, and reusable, which constitutes the FAIR data principles. PIDs facilitate the reuse of research data by enabling researchers to easily cite and credit the sources of their data and protocols. PIDs also make data more interoperable with existing and future systems, since the metadata associated with PIDs follow established metadata standards and enable information to be shared and reused without the need for transformations. And finally, PIDs make data more findable and accessible by ensuring that the underlying metadata is retrievable across systems, providing persistent access to published resources, even if the content has moved locations on the web. This concludes this portion of the webinar presentation. Thank you all for your time and attention. I'm going to pass it to John Graybeal from Stanford University, who will discuss semantic identifiers and other resource identifiers.
Thank you, Anya. Appreciate that. I'm John Graybeal. I'm the technical manager of the Stanford University contribution to the RADx Data Hub project. And I'm going to bring this out, zoom out a little bit on this discussion and talk about other kinds of identifiers, particularly semantic identifiers, but also really any what we call IRI. Next slide. The three objects that Anya talked about are listed here: the DOI for digital object, ORCID for people, and ROR for organizations. All of these have a similar format, what we would call the protocol, beginning with HTTPS://. And this is the protocol of an IRI, formerly known as URL or URI. You may have heard of it. You're going to get to hear a lot of acronyms today. Sorry about that. And it stands for Internationalized Resource Identifier. And it has the characteristics of being both resolvable if the user who provides it chooses to resolve it for you when you click on it, and being globally unique. IRIs provide a baseline for almost all digital persistent identifiers because they can be resolvable and are globally unique. Even if it's not resolvable, you can still use an IRI as an identifier, and that's perfectly legal. But it's not as fun from a social standpoint because you can't find out more information. Next slide. So IRIs have a lot of really good characteristics. They're persistent. They can last forever. The person who issues them has to intentionally do something to make them last forever. They have to keep a system up, but you can design the system to support an identifier that is an IRI forever. IRIs are recognizable by people. People can see one and say, I think that looks like something I put in my browser, and can go find out more information. And you can adapt the contents of them to express what you want people to see about your system and about your identifier. They're free. This is a big win with IRIs. Once you get a domain name, your website is at a particular domain, then you can create any IRIs you want, and it's all up to you how many you create, and no one will ever keep track or charge you for those. So that's a big plus. The IRIs can be branded. Your domain name that you've used for your IRI can make up a part of it, of course, and it resolves to a website or service that you've created, so that's good. And the information in it can be related to your system in particular. So it's really a flexible system that can be used to represent anything you want and even have some personal meaning for the work that you're doing. Next slide. There are some concerns. There are a lot of people who are like, oh, you wouldn't want to use an IRI for that because, and that they usually list one of these issues. I think all of these are less significant issues, but are important to be aware of if you're going to use IRIs. As I mentioned, if the user doesn't actually make them resolvable, if the person who has provided them, who has provided this capability, doesn't make them resolvable, if their domain goes away, then that would be awkward. It would still be a valid identifier, but it means you can't look it up anymore. In general, when people create IRIs, most of them don't care about them lasting forever or a long period of time, and they don't necessarily care much about their particular meaning. They may even hide the meaning of the IRI. And so people tend to think of IRIs in general that way, as opposed to the very targeted ones that we would be creating for these purposes. They're not entirely free because you have to have your domain name and you have to have somebody who's doing the resolving service. If your branding changes, and so you go from being International Business Machines to IBM, and you had a bunch of IRIs that were at internationalbusinessmachines.com, then you'd have to figure out a way to forward those. and move everybody off of those old identifiers. And you have to think about how you want to design the names so that they can be persistent. Many or all of these problems are true of other identifiers as well, including the ones that we mentioned earlier, but some of the decisions have been taken care of already. Next slide. So why on RADx do we care about identifying things, and what is it that needs identifying besides the three things that Anya just mentioned? All of these different things, I'm not going to go over them all in detail, but these different types of things are classic things that get identified with IRIs. Keywords like your science domains or the types of participants you have can get IRIs. Things you put in lists, your organizations or your facilities or roles people play. Medical terms and concepts are all very frequently identified with IRIs. Diseases, drugs, chemicals, organisms, even taxonomic charts, all can get identifiers. The sensors and processes you use, including all of the things in your wet lab or your dry lab, and the information you might describe in your publications. All of these different kinds of things are identifiable with IRIs. So you know what you might want to identify. How would you give them an identification? Let's go to the next slide. There is this resource called the Semantic Web, which is built around semantic standards and tools that were designed by a smart group of people, the World Wide Web Consortium, who helped define the specifications that everyone would follow. And these specifications were intended for creating and passing around information models. Simple examples of information models, like the chart you see here, simple descriptions of things, or vocabularies or thesauri, almost lists of terms, maybe hierarchical lists. In biomedicine, there are many more complex models that get created. Sometimes they may have 1 million terms or more, and they may be very complex webs of relationships and connections that can be followed and tracked. All of the models are formed out of a simple statement, and it has a simple form. It's a subject and a predicate and an object. And each of those things can be IRIs. Next slide. So in this one slide, I'll tell you everything you need to know about the semantic web to figure out how the whole thing works, if you derive it enough and intuit it enough. These subject, predicate, object things, statements, we call triples in the semantic web. This is part of the Resource Description Framework, or RDF. So I'm giving you the 1-minute RDF tutorial. If you want more details about any of this, there are a few links here that we can paste in and supply later as well. As you can see, the subject and predicate are always IRIs. The object can be an IRI, or it can just be a regular string, just regular text that someone has typed into a form or whatever. So when you express it as a triple, you just list these IRIs out. The examples for IRIs for a data set and for the predicate, the idea of being created by. And then in this case, I used the ORCID IRI for myself as an example that we've seen earlier. And that triple corresponds on the graph to something like dataset object created by John Graybiel object. So it's really simple. The whole concept is really simple, and we're not going to talk any more about it now. We're going to go to the next slide and wrap up here. Next slide. Thank you. So there are a lot of different things that we can start doing once we have everything defined as IRIs. We can precisely capture and look up knowledge, unifying how we describe vocabularies and more complex concept models. And we can make those descriptions available via computer tools and services. Computers can even add knowledge to these structures by performing logical computations on them, making inferences on them, and doing additional analytics. We can manage millions of concepts over time. So millions of terms, even billions perhaps, are already defined, integrated, referenced, and shared across different domains using this technology. And because they persist over time, you can come back and see what was going on and how these terms were originally defined. Resources like BioPortal and the OBO Foundry that you may have heard of, mostly focusing on biomedical terms in these cases, they can access both the information and analyses that are based on thousands of independent vocabularies and how they relate to each other. BioPortal alone, as an example, has nearly 15 million terms like this. So it's a very mature capability at this stage. You can build and reuse domain neutral tools because all of the ways that we structure these information sources are standardized. So tools like REDCap that you may have used often, or CEDAR that we're using a lot on this system, on RADx, can leverage the knowledge sources that have already been defined and can be updated automatically as the knowledge sources get updated. If you're familiar with the linked data community, the linked open data community, and all of the resources that it has, they also can take advantage of these structured resources. Finally, you can meaningfully share data and metadata. And so users can look up any of these terms. They are resolvable after all, or groups of terms, using either the identifier or even its label or description. Search tools like Google can know how to look up the pages that are in BioPortal. And when you search for these biomedical terms, often one of the first links you'll get is something from BioPortal that describes what that term means. And even automated systems can use the encoded knowledge to make data entry cleaner and more accurate and more complete, because it's looking up the very precise spelling and the very precise definitions, and users can take advantage of that. So I've described how using these IRI identifiers can make the world better and can make RADx better. And now you'll hear about a particular form of this identifier that can make IRIs simpler to use. So I'm excited to hear from Peter Rose of UC San Diego about CURIEs.
Susan, can you unshare your screen? I'll share my own. Okay, hello everyone. I'm Peter Rose. I manage the Structural Bioinformatics Lab at UC San Diego at the San Diego Supercomputer Center. I'm also a member of the RADx Data Coordinating Center. I'm involved with data harmonization. So since we heard a lot about ORCIDs and RADx, I thought I show my own ORCID and RADx here. And you see, I don't have the HTTPS, etc. in front of it. So I'm using CURIEs, which are compact identifiers, compact URIs or IRIs. So for example, you can write ORCID : and your ORCID ID or your raw ID and so forth. So those are CURIEs. Those are basically short forms that you can also use. So why do we need CURIEs or unique identifiers in general? And here are a few examples from my own experience. I recently wanted to look up all the publication for the RADx projects, and I found out there's actually a gene called RADx as well. So there can be some confusion. If we use unique identifiers, we can distinguish that, for example, for this gene, we could specify this HGNC identifier, and that will be unique. Here are some more extreme examples from the protein data bank. There's an identifier 1E03 or 1Fab, which are typically misinterpreted as 1000 or February 1. Actually, if you paste those two identifiers into Excel, it will exactly do that. It thinks this is 1000, this is February 1. And then the protein data bank also has small molecules called ligands, and they have a 3-character identifier, and like LAX, and that causes ambiguity. And it could be an airport code, or it could be a ligand in the protein data bank. Just for fun, I thought I asked ChatGPT, what it knows about LAX in the protein data bank. It says it's unlikely you find information about LAX in the protein data bank because it doesn't know the context. If you have LAX somewhere in text, it doesn't really know what it means. Another example here is from the CAC database I recently used. Their identifier starts with a C for compound and then a 5-digit number. And you can find it in many different places. Those can be, catalog numbers, can be a hexadecimal code for a color, or in our case, we're looking for the amino acid lysine. To be more specific, you could use a query like KEG compound identifier, or better yet, an INCHI key, which I will talk about later. Okay, so what are queries? They're basically compact IRIs or URIs. Basically, it's a shortened form that is more human readable, and you can use it somewhere in text, but you don't want to have a long URI. The general format is prefix identifier. So for example, DOI and then the DOI ID here. Those prefixes, they are registered namespaces. There cannot be anything here. They are a W3C standard, and they're also globally unique, and they're machine resolvable as a URI or IRI. Again, they're also a persistent identifier, and as the underlying data location changes, it will still be, pointing to the same data sets. So here's an example, and this example is coincidentally or not also about CURIEs itself. So how could you reference this paper? You could, use the link to the website, which is not a good idea, since that will be non-persistent. You can use an IRI like the DOI that you're familiar with. It's persistent, but it's long. Sometimes you want to have a real short identifier, and that's where CURIEs come in. You can then instead write DOI and then the identifier, which again resolves to the same IRI, but it's shorter, and you can use it somewhere in text, but you don't want to use an entire URL. Okay, what are examples of CURIEs? You can use it to reference publications. You obviously are familiar with the DOIs for publication. That's really the preferred way to reference publication. But in some cases, you may want to specifically reference a paper in PubMed or in PubMed Central. You could do it this way. You can reference any ontologies or controlled vocabulary. So this DOID, for example, is from the human disease ontology referring to COVID-19. Taxonomy refers to this SARS-CoV-2 virus in the NCBI taxonomy, or this MASH term refers to COVID-19 nucleic acid testing. And then, as I mentioned, for compounds, for chemical compounds, you really want to use an INCHI key. This is a different kind of identifier that can be computed from a structure unambiguously, and that's again the preferred way to reference compound. And this compound is actually a part of Paxlovid. And then CURIEs are obviously used, and that's what we're talking about today, for data repositories. There are generalist data repositories such as Figshare, Zenodo, etc. Sometimes GitHub is also used as a generalist data repository. You're all familiar with the Johns Hopkins COVID website, and they keep their data in this particular Git repository. Then specialist data repositories, they may have a registered name for the query. So for example, for dbGaP, there is the prefix dbGaP : and then the dbGaP session number. If you reference particular genomes, you can use RefSeq. For the sequence read archive, you can use this kind of query, SRA. If you reference specific proteins like the spike glycoprotein in SARS-CoV-2, you could reference the UniProt database using this, or you can also reference compound using, let's say, a PubChem compound identifier. So, and here's just an example. Here's the entire SARS-CoV-2 parts list, all its genes, proteins, and so on. And you can, every part on this picture, you can reference with a query. For example, here's the S gene, which you can specify by NCBI gene. This links to a GenBank. You can reference particular proteins like the spike protein here to this particular UniProt identifier. The three-dimensional structure is in the protein data bank with this identifier. Then you can reference the genome by a RefSeq number. You can specify the taxonomy, which is reference the SARS-CoV-2 virus. And finally, if you have a compound like this component of Paxlovid, which inhibits the main protease, you can reference by the INCHI key. So this way you can uniquely identify compounds, because, for example, if you just reference in text, you say spike protein or NSP1, that's actually not unique. Other viruses have the same kind of proteins with that same name. So this will be a unique way to specify all those components. Okay. So how do you know which curie to use? So there are registers for curies, and the first one is identifiers.org. It's maintained by AMBL, EBI. The European community has an effort called Elixir, and the identifiers.org is part of their interoperability platform. So they're really very much into interoperability in the biomedical space. So identifiers.org has curies for about 800 biomedical resources. And if you go to identifiers.org, you can search the registry and find the identifier that you need for the data set you are interested in. And to resolve a query, you prepend https identifiers.org in front of the query, and then it will resolve. So an example here, identifiers.org, and this is the query UniProt, and so forth. Another resource is Name2Thing, which is maintained by the California Digital Library. They basically include all of identifiers.org and a few more queries. So they have over 900 queries in general. And again, here it shows how to reference or resolve your queries with their service here. To just demonstrate what that means. So if you have this identifier UniProt PDB with this ID, and you prepend this, you click on it, it will resolve to the spike glycoprotein in UniProt right here. And similarly, the same thing will happen if you use the Name2Thing. Okay, so curies, they can support fair data management. First of all, they make your data findable because they use a unique and persistent identifier. They make data sets accessible because you can resolve them. And persistence ensures that you can access the data sets even if the underlying location on the web has changed. Makes it interoperable, and that's also very important for the RADx project. You can link heterogeneous data sets as long as they share a common queries. And finally, make research generally reusable because they unambiguously reference specific data sets or specific entities and so forth. So curies uniquely reference electronic documents, data sets, chemical or biological entities, oncology classes, or researchers, organizations, etc. And now in the area of generative AI applications like ChatGPT, this becomes even more important because you can provide a context or the ground truth, what each of the terms mean. So coming back to my example of LAX, ChatGPT thinks that's the Los Angeles International Airport. But if you would use a query to identify exactly what LAX is, that you don't mean the airport code, but you mean a particular ligand in the protein data bank, it would learn about that and know what the right answer is. So I think it becomes more and more important because your papers will be read as much by generative AI application as it will be read by other researchers in the future. So where should you use curate? In text, like method section, tables, or figure legend. Anytime you need a short identifier, you don't want to have a very long identifier with HTTPS, etc. Let's say in a figure legend, you want to have it short. In data files, databases, websites, web services. This was supposed to be a quiz. The obvious answer is, all of the above. And with that, I give it back to Rob Hubal, who will now share the Q&A session.
Great, thank you, Peter. Yes, so Robert Hubble here at RENC, and I'll serve as the host, so to speak, for our Q&A. I encourage anybody who has any questions to post them in the Q&A or chat, and we'll take them. First question I want to ask is, Peter, you were talking about this, and John, you hinted at quite a number of prefixes that are out there. How does one know about all those? Are you needed to know all those prefixes in advance? Or if you're searching, for instance, for a given gene, you must know that there's a certain gene database.
Yeah, so for example, you can go to identifiers.org and there's a browse feature. You can type in, okay, you want to look for the query for GenBank or something like that. You can find the prefixes there. That's the typical way you get those. Yeah.
So that's where you find the prefixes.
Yeah,
and from
that's
there
right.
you would know what to search.
Right, and they also provide you examples and so forth. Exactly how to use it. Yeah.
Okay. This question is for John, and Anya, more than welcome to participate as well. So DOI stands for Digital Object Identifier. Is it required that it's a digital object with some kind of URL or IRI that's associated with it?
So according to the Digital Object Identifier website and handbook, you can use a DOI for anything. And so that would be legal and fine. And some people do use it for other kinds of things, and there's nothing wrong with that. I think you find in certain applications, in particular in semantic applications, we don't see people using DOIs for concepts. So while in theory you could use it for representing terms the same way we use IRIs for representing terms and other things, it's a little less frequent. And I think part of the reason for that may just be that DOIs have a cost to generate that's used to maintain the ecosystem of DOIs. And so while it's a small cost, if you're going to be generating a whole lot of them, then you will tend to use something else because you don't want to pay for every single one that you're generating when it starts getting into the millions. Thanks for that question, Rob.
Sure. And Anya, do you want to? You had a suggestion as well about what the DOI stands for.
Right, and let me just restart my video. Yeah, so to John's point, DOIs could be used to identify non-digital objects, but in order to assign a DOI, there needs to be some sort of digital reference to that object, that non-digital object. So if it's, let's say, physical data, there needs to be metadata describing that data published so that the DOI can be referenced to some sort of URL for search and retrieval of information. But John is correct in that.
Great. Next question. Are there other repositories to search for queries other than the EBL that was mentioned?
Yeah, so the other one is Net2Things, which is maintained by the California Digital Library. So that's another way. But they're pretty much, there's a lot of overlap, almost identical. I see another related question there. If I want to create my own prefix, so if you have a data resource and want to create a prefix for that, you can contact identifiers.org or Name2Thing, and they will, talk with them, and you can create an identifier or a prefix for your data resource. Obviously, you need to be some kind of sustainable data resource that will have some long-term sustainability to do this.
Peter, one of these systems that I've used, and I can't remember which one it was, or maybe it was the third one, keeps track of what it thinks the recommended prefixes are by the number of people who have submitted the prefix as the definition. Are you familiar with that particular example? So it's a bit organic. It's not like there's one answer that rules them all forever. It can evolve over time.
Yeah, I know there are some folks they're trying to build their own query repositories, but in terms of identifiers.org, that's really a unique set. There's no overlap. I think it is, they curate that. You cannot have the same query for the same entity.
Okay, I'll just, if I may, I'll just mention here, Rob, that we have a funny situation in that we have several repositories of semantic resources anyway, and for various reasons, in a few cases, not tons, but maybe 3 or 5, the same set of resources has a different prefix in different repositories. And so there's a little bit of a inherent built into the system prefix conflict that is in the end only resolvable through everyone agreeing out of band, okay, we'll use identifiers.org, or okay, we'll use this other prefix repository as our defined thing. We, when we're dealing with this in semantic forms, it is always necessary to define the prefix's relationship to the domain or to the namespace at the beginning of the semantic resource, so that everybody knows how you're using that prefix. And that's the only thing that makes it absolutely clear from a global standpoint, globally unique standpoint, which prefixes go with which resources.
Mm-hmm. It sounds like there's a little bit of shake-up that goes on naturally, that until people get some kind of resolution to common ontologies. Okay, a question that we had posted earlier: if I want to uniquely identify a collection of resources on my web application and want something similar to a DOI, but don't want to pay for that DOI and associated, are there any suggestions?
Well, I have a few. One is that if you only want a few DOIs for a few resources, there are different places that will issue you a free DOI without cost, some library resources and others. I'm sorry, I don't have any that I can quote offhand, but I think you could probably find them through searching Google. I believe, I can't remember if it's Zenodo or another system, that if you publish your content there, then the DOI comes for free. And it's also the case that some library services will provide DOIs as a service, particular university library services. And so they're paying a cost, but as long as the number of DOIs being requested is not terribly high, then you can get free DOIs that way. And finally, of course, you can, there are other kinds of identifiers that you can mint, starting with IRIs if you have a domain, or other things like UUIDs, which are not resolvable, but are unique identifiers and are unique globally by virtue of how big they are, how random they are.
Okay, there's a question: are CURIEs useful for biorepositories, and how can they relate to materials across different RADx programs, such as the industry-targeted RADx Tech and RADx Advanced Technology versus the academia-targeted RADx Red and RADx Up?
Yeah, I mean, the CURIEs were originally developed, by Identifiers.org specifically for biorepositories, because that's EBI's business, in terms of relating materials across RADx programs. That is, let's say you're talking about a specific gene or specific protein or any other type of entities. If we have the same CURIEs, then we can link those data sets together. I mean, that's the idea of RADx, that we can mix and match the different data sets, see what they have in common, and that would be one way to do that. Let's say they use, you run some specific assay that uses particular substrates or specific chemicals and so on. That would be one way to link those materials together.
So let me clarify. So would you call common data elements CDEs? Do those typically have some kind of associated CURIE or IRI that is similar?
Yeah, we actually didn't assign them CURIEs, but we have specific columns. That is, for example, would be a protein data bank identifier like the UniProt identifier or GenBank ID and so on. So we have specific columns which are CDEs, and they are defined as being this particular, coming from this particular data resource. So we don't actually put the prefix in there because it's kind of implicit, because this particular column refers to, let's say, GenBank identifiers and so on.
Would it be helpful, Rob, if I just gave a 1-minute walkthrough of how you might go about building Curie-based CDE
Sure.
entry, for example?
Yeah, go ahead, John. Then we have one more question. I think we'll wrap up after that. So go ahead.
Okay. I'll make it a 20-second one then, so we have time. I think there's also one on the chat. When you have a set of resources like CDEs, if you register them in a repository, as has been done for some CDEs already in RADx and will be done for more, then you get identifiers that are full IRI identifiers. And you can create a prefix that can be used to shorten that whole path part of the identifier, the domain, and maybe even a little bit more. And once you've created that query and registered it, once you've created that prefix and registered it, then you can use it throughout to shorten that long identifier into something that is shorter and more manageable. And then you have queries for things. So it's not rocket science. It's a couple of discrete steps that are fairly straightforward once you're working with a lot of those things.
Great. Okay, Anya, last question, I think, is from Vivek. If an e-folder has many files, numerical data, text, images, do we assign the folder or do we have a DOI for each entity in that folder?
Mm-hmm. Right, so I was describing earlier how DOIs have an underlying metadata schema, and you can look online. Datasite, for example, publishes their entire metadata schema, but as part of the schema, you have to define what type of resource the DOI is being assigned to, and the resource could be like a data set, it could be a collection, it could be a book. But essentially what's important to think about at what sort of level and granularity you're assigning the DOI is how the end users, researchers are going to cite that resource in, let's say, secondary research. So if it makes sense to assign one DOI to the entire collection or to images or whatever is contained in that folder, because end users might cite those separately, that can be done. And DOIs, you can always establish relationships so that if a DOI is related to another DOI, that's specified in the metadata schema. So just keeping in mind what's most useful for the end user in order to cite that collection.
Perfect. So thank you to the panelists. I'm going to turn it over to Susan to wrap up the presentation.
Great, thank you all so much. So I just wanted to wrap up by mentioning that we're going to drop a link to the survey for this webinar here in the chat box for all of you. And just a reminder, you all, many of you are researchers and you all want your data and we want our data too. So please respond to our survey and give us some feedback. It really helps us better understand how it is that we can make these webinars even better. So please feel free to click on that link. And I wanted to mention thank you so much, Ari. Yeah, that's the link there for everyone for the survey. So I wanted to mention 5 resources for you from our RADx Data Hub website. We have our user guide, which includes step-by-step instructions for using the RADx Data Hub. So maybe you've never logged in before and you want to create your account and get logged in. So feel free to take a look at that user guide for step-by-step information. If you need a hand at any point, feel free to click on our help desk and we can lend you a hand as you navigate the RADx Data Hub. And we have a contact us form as well for additional support. And also feel free to take a look at our commonly asked questions there, the FAQs, and stay tuned for upcoming events. We hope to continue to collaborate with you all and share what we're excited about and what we're learning. So thank you all again so much for being a part of the webinar today. And on behalf of the RADx Data Hub Partners, wanted to thank you all for your participation. I'm going to share the slides and the recording of this webinar with the folks who signed up for this webinar in a couple days. Thank you all so much.